Hello and welcome to the talk about the research that we do here at Southampton. So the title of my talk is Making the Invisible Visible. And the reason I've called it this is because since I started my research journey, I've basically, when I look at everything I've done, I've really worked on imaging disease. Chemists are very, very instrumental in this process and the development over the years of lots of different imaging techniques. So I'm an organic chemist and I've been at Southampton since I started my PhD, which was too many years ago to mention, but a long time. I teach and I do research. So in this first slide, what you can see, this is one of our PhD students, Nikita, who's in one of our research labs, and she's doing a project where she's synthesizing small organic molecules. She's an organic chemist. And I just wanted to demonstrate how the students and the researchers are doing this organic chemistry, and this translates into the synthesis of molecules, which then translates into formulation of chemicals that can be injected into patients, which will then allow hospitals and such settings to capture images such as shown here of the skull, which can image all kinds of tissues, disease, tumors. And then obviously this then leads on to diagnosis, which is very important. So this process of going from a laboratory right through to the diagnosis of disease is crucial in our research. But what we work on is this part of the process. And today I'm just going to talk mainly on the work we've done around MRI imaging, and I'll touch a little bit on at the end about fluorescence in imaging. MRI stands for magnetic resonance imaging, which is basically the same as nuclear magnetic resonance imaging. And the reason that they dropped N from what would be NMRI is because in the early days of MRI, the word nuclear just frightened people. People thought obviously they were going to be zapped with some kind of nuclear radiation and they wouldn't want to go into the machine. So they dropped the word and it's just MRI. So really, when you're having an MRI taken, and I'm sure lots of students and yourselves that know of people who've had MRI scans, you're effectively taking a large NMR of your body, the human body, or part of the body. So MRI is an application of NMR for imaging disease. And what it gives us is it gives us a picture of all the soft tissues in the body and also things like tumors, so we can see areas of disease. And it works by taking an NMR of all the water molecules in your body. And it works quite well and it gives good images, and obviously it's used in hospitals all over the world. So you might then ask, but why are we researching in that area? Well, things can always be improved, and that's what we look at. So what's the problem? Well, if you think about it, water is everywhere in the body. There's a huge concentration of water in your body. This table sort of shows you the percentages of the amount of water in your body. So if you're imaging the NMR of water, there's loads of it. And it's quite difficult to see the difference between different tissues when you've got that much water in the body. So you're not going to get very, very high contrast images between a tumor and a non-cancerous tissue, for example. And also, when you're talking about very, very small amounts of cancers in the early stages, you might not be able to detect it. So what's the solution to this? Well, as organic chemists, obviously, we like making small molecules. We like making organic molecules. That's what we do. So is there a way that we can make other molecules that can be given to patients and that we can trace them? So we can make a small organic molecule, but it has to contain some kind of signal, some kind of trace of a part of it, so that we can see it in the NMR machine, in the MRI machine. Okay, so one way to consider looking at this is we think about the fact that tumor cells have an increased metabolism. So tumor cells will turn over glucose or break down glucose at a much faster rate than a normal cell. And this differentiation between the metabolic rate of a tumor cell and a normal cell can be harnessed in such a way that if we gave our patient maybe glucose that generated some kind of signal, or one of the breakdown products of glucose. So you can see here pyruvic acid, and pyruvic acid is a metabolite of glucose. It's a breakdown product of glucose, which then continues to be broken down further. So if we can make small organic molecules of this type that actually contain some kind of tracer molecule, then maybe we can see the difference in metabolism between two types of cell, and that will help us to detect the cancer. So pyruvic acid is one example of a molecule that's been worked on. The trouble is, if you give pyruvic acid, as it's drawn there, to a patient, all the carbon in it is C12. So you're familiar with the natural isotope of carbon that is C12. The trouble is, carbon is everywhere as well in our body. Every peptide, every sugar contains, we are made up of carbon. So if you give a patient a molecule like that, how are you going to see it? Because it's just going to disappear into all the signals of the rest of the body. So what we can do as organic chemists is we can make molecules that are isotopically enriched, as we call it. So basically, instead of having C12, we have C13 in one position in the molecule. Now, C13 is naturally abundant, so there is a small amount of C13 in every normal organic molecule, and that's why we can do NMR in the lab. But the level of C13 is only 1%, and that would not be strong enough signal for us to be able to detect it in a patient. So if we make molecules that have got C13 enriched, so they're 100% C13 in one position, then we can now detect it in the MRI scanner. And so we've done a lot of work on making organic molecules that contain isotopes of C13 to be able to detect them. And through chemical synthesis, we make these molecules as organic chemists. And we work with our collaborators who are physical chemists, who then develop advanced NMR techniques. So they advance the physical side of the chemistry to be able to make, and then when we work in combination, we've ended up being able to make signals that are 20,000 times more strong, which you can imagine when you're trying to detect very low levels of cancers, this is very important. So that's what we've been working on. But can we take this further? Can we improve this more? And which is something as researchers we're always thinking. Now, pyruvic acid, like I mentioned on the last slide, and water can both be detected by MRI. But the problem is with both of them is that the signal that is generated has a half-life, which means that the signal that they give out decays to 50% in a matter of time. And with pyruvic acid and water, this half-life is a matter of seconds. So the time you've got to actually take images is very short. And what we'd like to do is be able to extend this half-life. And we'd like to have a half-life that was much longer to allow us to take lots more images over a longer period of time and get more clear, distinct images. The problem with having a very short half-life also is that you can take multiple scans, but then that requires the patient to keep very still. And I'm sure if you've had an MRI, you've been told, don't move. You know, if you every time they move, and if you're taking multiple images, you're going to get blurring of your images. Whereas if you've got a molecule that can last for a long time, you can just take one distinct image. So one of our more recent publications is this molecule, which is quite a complicated molecule to synthesize, as you can imagine. It's based on a naphthalene bis-aromatic core, and it incorporates two C13 isotopes. And this molecule now has a lifetime in NMR of over an hour, which is the longest lifetime recorded to date. And we continue to synthesize molecules as chemists to try and extend this lifetime and to try and make molecules that at the same time having a long lifetime also have a biological compatibility. Because to make something that you can inject into a patient, there are lots of criteria, as you can imagine. They have to be water soluble, they have to be non-toxic, and they don't have to lead to toxic byproducts. So there's lots of things to think about when we do these research projects. And that's why our research projects last quite a long time, as you can imagine. So that's a little bit of work that we've done in the area of MRI. And that's really thinking about how to image disease in a patient who's in the hospital. But what if you want to image something different? What if you want to image infection, for instance? And then this is where fluorescence can be really, really useful. So fluorescent molecules tend to be highly conjugated aromatic molecules. So you can see this is an example of a molecule called fluorescein. It's highly fluorescent in that it generates a bright signal, and it is so fluorescent that you need a very, very small amount of it to be able to detect it. So you can see these cells that have been labeled fluorescently, they're very, very bright. So fluorescence is a very useful technique for detecting very, very small amounts of biological material. And where this is really relevant at the moment is in development of testing. So coronavirus is a very typical example. We all know the importance of testing for coronavirus. We've all been involved with getting tested. We know about the PCR test. Now, where did the PCR test come about? How do we know how to test? Well, these tests were developed originally by chemists, and we've done work at Southampton to develop some of these tests. And one example of this is in using synthetic DNA. So we make DNA that contains a fluorescent label. And so typically how a PCR test for COVID might work is that when you give in your sample, it contains obviously your DNA, but if you are positive for COVID, it will contain a small amount of the viral RNA, because COVID is an RNA virus. So it contains a small amount of this RNA from the coronavirus. And then what PCR does is called, PCR stands for polymerase chain reaction. And it is basically a technique that amplifies the amount of viral DNA that is present in the sample. So it just makes lots and lots of copies of it, because the amount of viral RNA that is present is so small, it's very difficult to detect. So the first stage in the PCR test is to make lots of copies of that RNA. Then when we have lots of copies of the RNA, that's great, but we still can't see it. We don't know it's there. And this is where our imaging comes in. We as chemists make synthetic DNA, and that's actually made here at Southampton by a company in the building. They make the DNA and they incorporate fluorescent groups. And this example you can see here is one type of what we call a DNA probe that we synthesize. The DNA probe has this hairpin type structure. And this is a sequence of DNA that is complementary to the viral RNA. And when I say complementary, I mean it base pairs. So at A level, you will have done A, T, G, and C. So if you have a strand that is labeled in such a way that is complementary to the RNA of the virus, in the presence of this RNA virus, it will bind. And it is such that when this binding occurs, this sequence in the loop will bind, and this separates these two groups. So these two groups are chemically synthesized small molecules. This is a fluorophore, a fluorescent molecule, and this is a quencher. When these are in close proximity, the fluorescence is quenched, and this is a dark probe. There's no signal. It's a negative signal for the PCR test. But when it comes in contact with the COVID RNA, it binds, and this breaks this short stem, and this part of the molecule binds the RNA, and then the two molecules, the fluorophore and the quencher molecule, are separated. And now we have a fluorescent signal, and that gives us our positive reading for a PCR test. So this is one of the ways that we are all being able to be screened for COVID. And so you can see the importance of the organic chemistry that we do, and the importance of chemistry in the diagnosis of disease. And that's the end of my talk.